Let me start with plant health because my field is plant health. I just want to set the stage. I know not everyone works on plant health, but I still want to set the stage around that. But honestly, anything related to plant biology or microbiology would be relevant here. I think the new thing really is that this problem of plant disease and plant health has been addressed with chemistry for a long time. Fungicides, chemicals, etc. And right now, there is a sense that there is an opportunity that biology can progress fast enough to keep up with pathogens and disease. This is something that is new. It's not fully established. I think this is work in progress, but there's definitely in my circles with the people I talk to are both on the fundamental and applied side. There's a sense that biology is kind of catching up to really start advancing at a fast rate, similar to chemistry, to be honest, and at a rate that can keep up with pathogen evolution. And because of this, there are really huge opportunities in biology, in plant pathology, in plant health, in plant biology at the moment. Maybe opportunities that were not there just a few years ago. The reason, in my view, this is an analysis I did for the FAO recently. They asked me to kind of sort of reflect on what are the opportunities in the field of plant health in the next 5 to 10 years. And what I came up with is this kind of three pillars, and this is really one inflection point. They're all converging at the same time, which is quite exciting for us. The first thing is genomics. And genomics, I think you all know what this is, but this is really more about knowing what we're studying in absolute details. In the past, I used the metaphor of a book. If you don't have the genomes, like studying a book without opening it and reading the text. Genomics really is becoming more and more accessible, and this is what's allowing us to really know exactly the crops we work with, the pathogen we work with. We have all the genes, all the list of genes, etc. Now we need to just put those puzzles together. The second thing is the AI input. And AI really sometimes is misunderstood because what AI does more than anything, it accelerates the research. This is very important. Very often people compare AI performance to human performance, and that's not the point. The point is, even if AI is performing as well as humans, not better, or even if it's performing a little bit less efficiently, it still can accelerate the research because it can be automated, can be scaled up. And that's really important use of AI. We can now accelerate the science we do, we can shorten some of these experimental cycles, etc. And the future really of AI is that we stop guessing, we start making better predictions. And really, a lot of things we do in genomics, once we sequence the genome, is making predictions. We predict which parts of the DNA code for genes, which ones are exons, which ones are introns. Uh, we predict what the protein sequence is. These are all predictions, right? But now we can actually push this forward all the way to protein structure. And that's actually the main topic we will emphasize in these workshops. So that's the second pillar, if you like. And the third pillar is the changing regulatory environment. And maybe in Tunisia, this is a little bit fuzzy, but in UK, in Europe, this is really changing very rapidly and has changed recently. The UK has now regulated lightly gene editing as a different technology from GM, from OGM and transgenic plants. So gene editing now is quite lightly regulated. We actually have some field trials here in the Sainsbury Lab that we'll be doing on potatoes and tomatoes in the coming year with GE or transgenic plants. Sorry, cisgenic plants, not transgenic. And because of this, capital investments is following. And also the EU, even the EU, which is traditionally very conservative, has recently agreed to regulate these new plant breeding technologies, including CRISPR gene editing, lightly. So there's a lot of opportunities, and because of these opportunities, there are some investments that are following. So this is huge opportunity also for markets like Tunisia and other parts of global south that export to Europe and so on, because now this is something in the next 5 to 10 years we can consider being a product that can even be an export product. Okay, so I think this is exciting because these are three completely different things that are converging at the same time. And with James, James and I, we've been discussing about these issues for a while, and the first pillar is really genomics, and we realize that genomics is not that accessible to the global south. Many reasons. I don't want to discuss that today, but I think you yourself know what these reasons are. But it's not just cost; it can be more complicated than that. But we decided a few years back to put together GetGenome as an independent nonprofit NGO to basically provide genomics to the global south. We started with Tunisia, but now we are actually present in many countries, as you can see on this map. And yeah, GetGenomes has grown to the point that now we are not only sequencing genomes, but also assisting our communities to interpret and make the most out of these genome sequences that they have. And this is also the context under which we are doing this workshop and the September workshops. And the concept of GetGenome, maybe you heard this before, but I want to really address it again because I think it's so important, is that a lot of people think of genomics as the process of sequencing genomes, assembling them, annotating them. What we want to do with GetGenome is we want to like take away that boring, tedious routine work from you. We want to just immediately go from your samples to give you annotated genomes that you can already analyze. And that's really where the real work starts, right? The point of like sequencing, assembling genomes is just routine work that is actually these days done by companies and by service providers. We don't even do it here in our institutes, in UK or in other advanced institute. We don't really bother to do these steps ourselves. We use service providers. And so why should you in Tunisia or in other countries get stuck with this? And as you know, this can be also tricky and complicated, technically um, tedious, and requires some technical know-how that's not very useful unless you work for a company or sequencing service. Then it's worth it. But in many cases, you really don't want to bother with that. You just want to have your annotated genome given to you, and then you can start the real work of extracting as much biological information as you can from that annotated genome. So that's our concept in with GetGenome is really the to leapfrog this routine work that we don't even bother to do here in our institute. So why should you? And as you know, this is also done in the context of very serious stresses on agriculture, on our societies. Of course, drought. You're now having this heat wave in Tunisia, which is really extreme. And this is actually a slide that was sent to me a few years back by Dr. Mazouri in Kef, and he was doing his barley breeder, and he was doing some field trials in Kef. And when he planted in January, there was no rain for a whole month, and he lost all his populations because of that lack of rain. So this is really dramatic. I mean, this is having impact not just on agriculture, but also on research and breeding in enabling people to deal with this very unexpected patterns of rainfall and heat and all of that, right? And of course, now I'm sorry to show you this slide and remind you that you are suffering from this terrible period of extreme temperature. And actually, it's really shocking how divergent it is from the averages. And this is a global problem, but it's affecting some regions like North Africa more than others. So this is also part of the concept of GetGenome and also part of our concept of working with the Global South, because you really should be the ones identifying the material, the biological resources you have in your countries to prioritize for genome sequencing. And so this is the project we did with the Durham GPT network in Tunisia, and they identified these two varieties. Shili and Mahmoudi, and we prioritized them for sequencing. But this was really bottom-up project, and I think just that aspect, the fact that this came from your community, the Tunisian community decided this should be the highest priority, is a very original aspect of doing this research. And the project was totally owned by the Tunisian scientists, and now it's at the stage where how to interpret these genomes, how to extract the most information from them. And of course, the idea here is that these are varieties that are adapted to more extreme conditions, and hopefully they'll be carrying genes and traits that are resilient and useful for wheat breeding and also useful for advancing the research. Okay, any questions so far? Any comments? No? Okay. All right, so now we sequence genomes. We have microbial genomes, we have durum wheat genomes. What do we do? What now? So this is the typical kind of um, computational analysis, usually computational, that are done once we have genome sequences. And again, I want to emphasize that a lot of these are predictions. Sometimes we forget that we are actually making predictions with a degree of uncertainty. It's never a completely accurate exercise. And that's, today, if I have one lesson for you to really take home and keep in mind as you proceed, is that all of these analysis, all this computational analysis, whether it's gene prediction, predicting SNPs, comparative analysis, etc., all the way to AlphaFold and predicting structure and all of that, these are all computational analysis that come with uncertainty. There is always a level of error. It's inherent in any computational analysis, never perfect versus imperfect. So never think about in a binary term like this is a correct or incorrect prediction. It's always coming with uncertainty. Even, for example, predicting gene models, predicting introns and exons, it can be very accurate, but we also know that there's a biological phenomena of alternative splicing. So even if your prediction is accurate, that doesn't mean it explains the full biology, right? In the same way, if you're predicting protein structure, that protein may exist in complex with different proteins and may also change its structure depending on which protein it's in complex with. And some proteins also oligomerize and all of that. So all of these are predictions that come with some uncertainty. And it's very important to keep that in mind. SNP calling, for example, we often forget is also a very uncertain exercise. We have, for take wheat, it's a hexaploid, so it's very complicated to call SNPs because SNPs can be present in different level of basically of ploidy. So you could have a SNP that's present only in one out of the six haplotypes, and that will be maybe more difficult, more challenging to identify compared to SNP that is present in all six haplotypes, right? So you have heterozygosity, you have all these kind of issues. So it's very important to keep in mind that we have a degree of uncertainty always associated with predictions. And what's important from your perspective to understand what can be causing errors and also what is my degree of confidence in this analysis. And that can vary depending on what you want to do with that analysis, right? It depends on the downstream analysis. For example, if you're calling SNPs and you want to use them as markers, maybe you don't want to miss any SNP. So maybe in that case, you don't mind if you have 20% false calls, because then you capture everything and you're not going to miss the important SNPs that are linked to your trait. But if you're calling SNPs to, for example, I don't know, find mutants in proteins, then you want to be maybe more accurate and reduce the sensitivity, but increase the accuracy, right? So this is a very important aspect of this computational analysis that you need to keep in mind. And sometimes when we talk about AlphaFold, you'll hear a lot about discussion about how confident the model is, etc., etc. But this is really true for everything else you do in bioinformatics. Okay. All right, so these are the different things we can do in once we have a genome sequence, and what we will emphasize is three different aspects. Is one is structure modeling, which is I will talk about a little more later, and this is basically predicting a protein structure only from the sequence, solely from the protein sequence. And the second aspect are the binders. So this is basically making synthetic proteins that bind your protein. And I'm not going to talk about that today, but there will be some of my colleagues who will talk to you later will talk about that. And the third aspect, which I find incredibly exciting, and it's really in the very early stages, so I wouldn't call this a kind of a fully solved problem, is to predict protein-protein interactions computationally. For example, if you start with a pathogen genome and a plant genome, can you predict all the interactions between pathogen proteins and plant proteins only using computational methods like AlphaFold? So this is really exciting because what this does really, it allows us to get even more information out of the genome. And I'm really, really excited that in that leapfrogging exercise we talked about, I can take your community all the way to using these really state-of-the-art tools like AlphaFold and so on, and even getting you ready to these protein-protein interaction predictions, which I think are going to be completely game-changing in biology. So that's really the logic and why James and I and our colleagues are really motivated to give you these workshops and introduce you to these methods. Okay, so yeah, these are the topics, and I don't need to go through this. James showed you this last time, but these are the different talks we'll have from people who are really using these tools. So it will be from experts already using and quite familiar with these different tools. Okay, so now that was kind of an introduction I wanted to give you, but now I'm gonna delve into the specific examples I said I will cover of applications of these AI tools to our own research. So my research, the research in my lab is on plant immunity. So it's really the business of plants having immune receptors that detect pathogen molecules. So we have cell surface receptors like the ones here, the so-called PRRs, and then we have intracellular receptors, the so-called NLRs, that detect pathogen effectors that are delivered inside the plant cell. And if you are coming to this from a plant breeding perspective or plant genetics perspective, usually these receptors are coded by what we call R genes, resistance gene, disease resistance genes that basically code for receptors that can detect particular pathogen proteins. So our research topic is around this, and the examples I will give you are around this business of immune receptors detecting pathogen proteins. When we sequence genomes, we have basically an alphabet soup of genes, right? You have a list of genes, thousands of them, and really the main exercise we do is making sense of this alphabet soup, right? So a very important aspect of bioinformatics, of exploiting information from genome sequences, is to make sense and organize this alphabet soup in different categories and hopefully connect even things together. So if I want to kind of summarize a lot of the things you would want to do with these weird genomes and the microbial genomes is really making sense of a gene alphabet soup. And of course, depending on your interest, you would be interested in different types of genes and different categories of this alphabet soup. Of course, in our case, we care a lot about the immune receptors. In particular, we care a lot about the NLR, intracellular immune receptors. So I will give you examples around that. But conceptually, this applies to any other gene family, any other protein family you would be interested in. You might be interested in transcription factors, in kinases, what have you. It's conceptually the same challenges. And the way we approach this challenge in the lab is very simple. We have three different activities that are complementary and that are actually feeding into each other. One of them is phylogenomics, which is basically taking these genomes and organizing, extracting protein families from that, and then organizing them based on the evolutionary history. So classifying them based on evolution, which ones are more related to each other, can we identify how many different subfamilies we have, etc., etc. The second one is structural biology, which can be experimental, of course. We can solve structures of proteins experimentally, but of course, these days is assisted by AI systems like AlphaFold, where we can predict the structure of proteins. This is also useful for classifying them into categories. It's more sensitive than approaches using only sequences, but it's also useful for predicting function, predicting localization, even predicting, like I said, protein-protein interactions. And so these two activities, structural biology and phylogenomics, These days, they are computational, but they're very much AI assisted. So when we talk about AI impact on biology, these are two fields that have been massively impacted by AI. And the reason is that this is really big data analysis, whether it's phylogenomics, predicting genes, annotating genes, etc., building phylogenetic trees. These are all approaches that can really benefit from machine learning and AI type systems. And there have been quite a number of systems that have been developed to facilitate this. In the same way, we'll talk more about that later. Structural biology, of course, with AlphaFold in particular, has been very much impacted by AI. On the other hand, at the moment, the functional analysis still require humans. We don't have robots to do the experiments, not yet. Maybe it will come in the future, but this part, the functional analysis, is still human-driven. I would say we don't really have any AI input at the moment into that aspect. But of course, the results from these experiments can feed into our knowledge and our AI systems and help improve them. Okay, so what I want to do first, I'm going to tell you a little bit more about phylogenomics and this first part, because I think that's an important aspect that you need to really, I think, give some attention to. Of course, we are assisting you, and your collaborators also are assisting you with annotating genomes and all of that. But there are challenges that can be related to this that I think is useful for you to be aware of, even if you're not doing the gene annotation yourself. So not long ago, actually, when was this? June 23rd. So like a few weeks ago, my colleagues and I posted this blog article on gene annotation, on genome annotation. It's called No Mokusatsu in Our Genomics. And the article was written because actually in our team, we bothered to re-annotate a lot of genomes using a tool called Helixer. Yeah, so Helixer is an AI system that can annotate genomes. It's not perfect. None of the genome annotation systems are perfect. But Helixer has advantages in the sense that it is very well trained on plants, on plant genomes. Actually, was developed by a group that primarily worked on plants, so it's quite good in our view on for annotating plant genomes. And also, it's relatively fast and efficient, so we can really re-annotate hundreds of genomes. And the reason we bothered, so we really wrote this blog because some people are puzzled. Why are you bothering to re-annotate genomes that are already in NCBI, already annotated, right? And the reason is this mokusatsu kind of inspiration, if you like. Okay, so what's the story with mokusatsu? So mokusatsu is a Japanese word that was very famous during World War II. Towards the end of World War II, the Allied forces, so the US and so on, they offered to Japan the opportunity to surrender. And the Prime Minister of Japan at the time, he replied with one word. He said mokusatsu. And the problem with mokusatsu, it has two meanings in Japanese. One meaning means to ignore, even ignore with contempt. Another meaning is to wisely stay silent. So they are completely different meanings, right? And historians are still debating this, but the Allied forces, the Western forces, they interpret this as basically a rejection of the offer made to Japan. And not much later after that, there was Hiroshima and Nagasaki bombs and all of that. So they took this as Japan said no. But actually, some historians said that Japan didn't say no, we don't want this. They just said we want to think about it and stay silent for now. So that's my point here is that one word, one gene, can be annotated completely differently depending on how you translate it into something else. So that's what our inspiration here was about. So a piece of DNA, you could interpret it as I have this gene here, or you could interpret it as I have a different kind of meaning. So that was the idea, no mokusatsu in our genomics, and that's why we bothered to re-annotate all these genomes with the same tool. Another reason was this paper that's very influential, and it showed that if you are looking, for example, for lineage-specific genes, so these will be genes present in certain species, but not others, right? Like let's say you are looking at grasses or cereals, you may ask the question, which genes are present in wheat but missing in barley, for example, right? And what they showed is that if they use the public annotations, they find a lot more genes than in reality. But if they harmonize the annotations, they use one annotation tools across all genomes, then the number of linear specific genes drops dramatically. And they did it for fish, cichlids, and primates, and they had the same results. And these were based on actually published papers. So, one-fifth of the papers were wrong in reporting so many genes as being linear specific, whereas it was not linear specific, it was lost in translation type problem, right? And you can imagine why that's the case, because if you think about genome annotation, a lot of the reference genomes we have are very well annotated. There's RNA-seq information, etc., etc., but maybe other genomes are not as well annotated. So you will have like an imbalance in how well annotated different genomes are, and therefore your analysis could be misled by that differential in annotation rather than a difference, true biological signal, right? So anyways, we bothered to do this, but because we wanted to really make the community very much aware of this problem. But also, this is also our justification for doing this, which is what we call harmonized annotations, where we went back to the genomes we study and the plant species we study and re-annotated using one tool, Helix. It may not be perfect, tool for genome annotation. No tool is perfect, but at least now it's harmonized and we can give more confidence, have more confidence when we find differences between the genomes we study. Yeah, this is what Amir Ali and you did, and they will be talking to you later in this program, so you'll hear more about that. But one project, they had several projects. I think they annotated now maybe close to 1,000 genome, re-annotated close to 1,000 genomes, including plants and fungi. But in this particular project, we focused on Solanaceae, so the potato tomato family, and they re-annotated 350 genomes that are publicly available that were also selected as being high quality. So they had the quality QC, if you like, filter to eliminate the genomes, the assemblies that were not very high quality. Out of those genomes, they have 15 million proteins. And out of those 15 million proteins, there was 200,000 immune receptors of this NLR family. But the point here is that this is like a really good data set because they're all again, annotated with the same tools. And also, they're all formatted in exactly the same way. So it's much easier to extract information out of these 15 million proteins versus like if you have download them from NCBI, some are formatted in one way, some are other, etc. So it's again, not only is the genome annotation harmonized, but also the data set is harmonized and the data formatting is harmonized. So it's very easy, for example, for them to go and extract all the proteins out of the 50 million that have a certain domain. For example, you might be interested in, I don't know, MADS transcription factors or something like that. And there are interpro domains associated with those MADS transcription factors. So each one of these 15 million protein has all the interpro domain associated with it. So it's very easy with the script to quickly pull out, I don't know how many it will be, maybe a few 100,000 proteins that have that particular domain, right? And so this is the value of investing time and energy in creating high quality starting data set. Actually, I like to call it genome curation, if you like, because you're now curating a data set that's very high quality, adapted to your project, to the biological question you want to ask. And I think this is something that your community can really invest in because once you have a data set like that, it's an incredible resource. Can you imagine these 15 million proteins from 350 tomato, potato, etc., solanum genomes? Like how many projects you could be initiated out of this, right? It's quite incredible. But you'll be talking to these experts, so feel free to challenge them and ask them more questions about this type of analysis. And then the other thing is the first thing to do once you extract your family, like in this case, we have 200,000 immune receptor of this NLR family, extracted, by the way, using interpro domains and using very simple scripts where you decide what are the features of these proteins, and then you simply extract them from the 15 million. So that's 1.3% of the total data set. It's quite a large number, by the way. The plants really invest a lot in immunity. And then they can build the phylogenetic tree we can build these trees with 200,000 proteins. This is possible these days. There are tools again to enable that. And now you can organize them in different categories, different families, what we call clades, phylogenetic clades. And yeah, and then basically you can organize your data through this phylogenetic framework and start asking a very specific question about your data set. For example, you could color code them by species, you could color code them by presence, absence of certain motifs or certain features, etc. And that a fantastic framework to start doing the protein structure modeling and looking for differences between different families and their structures and so on. But I want to emphasize this part because I feel the curation of the genomics data we have and this type of harmonized um, annotations and harmonized data sets is not respected enough. And that's why we wrote the article. And I see a lot of papers being published, even from leading labs, where they don't bother to do this. But again, I want you to go back and look at this paper and really give some respect to the fact that if you just download data from NCBI and start building stories around it, you might be losing something in translation there. So I want to emphasize this point. It's not necessarily an AI assisted application, although Helixer is an AI system, but I think it's something I want to really emphasize. And this is something that maybe, I'm not sure again where we'll be at during the workshop. Maybe you and Amir Ali will have already data sets, like maybe they'll have a few hundred serial GRAS genomes already reannotated that you could use as starting point when you meet them at the workshops. Okay. Oh, yeah. The other aspect of this is we actually do publish this data. So all these harmonized data sets, majority of them are published. We use the Zenodo community. So we have this open plant and LR community, for example, where this data set can also be published as data sets and shared with the community. These are under DOI, Digital Object Identifier, so they can be cited, etc. So this is something I think you've been exposed to before, but the point is like we can also share these data sets. I think they have value, and hopefully several labs can start using them. So now I'm going to shift gears to AlphaFold and give you the three examples I promised to give you. And these are three examples from our own research, very recent examples on how we applied AlphaFold 3 in particular to studying these immune receptors. And one of the shocking thing, and really still shocking for me to say today, is that AlphaFold 3 only came out 2 years ago, a little over 2 years ago, right? And there's so much happened since. I really feel like it was 10 years ago. And one of the things that happened, of course, is also that 6 months after this, in November 24, October, November, I don't remember, but uh, the DeepMind team was awarded the Nobel Prize of Chemistry for the discovery of AlphaFold. So this was actually incredibly shockingly short period between the emergence of AlphaFold as a tool and the Nobel Prize awarded to it. So that in itself tells you how much impact it had on biology. So I'm going to talk to you about the immune receptors, and I need to give you a little bit of background so you can appreciate what I will talk about. In the lab, we study how these immune receptors get activated. So plant immune receptors of the NLR type, they are switches. They sit in the cell in an off state, and then they get activated by when pathogens come, they get activated. And one thing we know is that in many cases, the off state is a dimer, a homodimer, that gets activated by releasing the two moieties of the dimer. So actually, I have this model right here. Maybe I can show it. And so this is the homodimer, and it activates simply by going like this and opening up. And then it's a very easy model to test experimentally because if you make mutations in the interface, so the interface is the yellow part, if you make mutations there, then the protein becomes autoactive because it cannot stay auto-inhibited in a resting state. And so it's a very straightforward model. But once it activates, each one of these protomers go on to form what we call a hexamer. So the hexamer is here. This is the hexamer. So this is six of them coming together and forming this oligomeric ring-like structure. And this is actually a channel. You can actually see through it. And this is a channel that goes into the plasma membrane and function as a calcium channel. There's probably other activities that we don't know, but at the moment, this is known as a calcium channel. So the point is that these proteins again activate from this homodimer state into this hexameric state. I'm going to show you the channel here. I have a video. It's worth seeing. And in this, this is actually also an AlphaFold 3 model. We put these little white bars here are actually oleic acids that are mimicking the plasma membrane. And you can see that AlphaFold is even placing the phospholipid exactly where they should be in in the funnel. So that funnel is what inserts into the plasma membrane. So this particular funnel here is what goes into the plasma membrane. So my fingers would be the plasma membrane. This inserts there and functions as a channel. Okay, I'm telling you all this because this is important for the AlphaFold application to studying these proteins because basically 2 years ago we were writing a paper on a structure, this structure of the hexamer. We solved it experimentally, 2 years of hard work of purifying proteins and everything and so on. And after solving it, we start writing paper, of course, right? And while we were writing the paper in May 24, AlphaFold 3 came online, and we realized that AlphaFold 3 is predicting perfectly the structure that we just solved, even though it was not public yet, right? So it was actually quite an interesting moment in the lab because some people were depressed and were like, "Oh my God, like anybody in the world now can, with press on a button in a few minutes, get the same structure that we worked so hard to solve." So it is actually a really interesting moment. And what you see here is on top is the cryo-EM model, and below is superimposed the AlphaFold and the experimental model. And this number here, RMSD, 1.7 angstrom, is actually the distance between the atoms, average distance between the atoms in the experimental structure and in the AlphaFold model. And that number is really, really good. That's very, very close. So usually when it's below 2 angstrom, that means it's more or less the same structure. And so that means that basically the two structures, the AlphaFold one and the experimental one using cryo-EM technology were very similar. So it was a bit of an interesting moment, and we decided at that time to basically include, actually take advantage of this and include the benchmarking of AlphaFold in the paper. So the paper, half the paper became about AlphaFold is really good at predicting the structure that we just solved and using our structure and two others that were previously published. So, to cut long story short, in the last 2 years, it's been crazy because this has really accelerated research on these proteins. And we have published already many papers that I'm sure my colleagues will tell you also more about, because it just became like just crazy. Like all of a sudden, you have this tool that allows you to predict these structures, which we call resistosomes, by the way. I think I didn't mention that, in many different ways. And so I'm going to tell you now three applications of AlphaFold 3 to studying these proteins. The first one was in collaboration with my colleague Torga Boskurt, who's at Imperial College. And Torga's lab has already made an observation before AlphaFold 3 came online. And that observation was that one type of these NLR receptors, the so-called NRG1, which is shown here on the right side, NRG1 is they did some confocal microscopy, some cell biology, and they found that NRG1 accumulates at chloroplasts at the organelles. And at that time, this was a very strange result because everybody expected this resistosome to go to the plasma membrane. So what is going on here? Why is this protein not going to the plasma membrane or going in addition to plasma membrane, going to the organelles? And then when AlphaFold 3 came out, it predicted this really strange, elongated funnel of NRG1 compared to the others, like NRC4 will be similar to the one I just showed you. ZAR1 is a canonical one. So these are like the ones that are known to go to the plasma membrane, and they're basically half the length of NRG1. And the reason is that NRG1, that extended funnel, is actually long enough to span the double membrane of the chloroplast. You know, organelles are derived from bacteria, so they have two membranes, unlike the plasma membrane of a plant cell. So they have the outer membrane, the inner membrane, and that elongated funnel of NRG1 is sufficient to span the double membrane and therefore would be different from the typical plasma membrane associated funnel or resistosome. So when AlphaFold 3 came and Toga made this observation, then they gained a lot of confidence in that observation they had before that NRG1 goes to the chloroplast. So they went back to their old data and basically revisited that data using the model, the AlphaFold model, used the model to make mutants, etc. And now it's all published. But that was like essentially an incredible unusual atypical observation that was independently validated by AlphaFold. So this is also how AlphaFold and this modeling can be helpful in giving you support for some observation you may have made experimentally. Okay, so that's one example. And yeah, so I forgot about the slide, but basically the model now that they present in the paper is that a typical NLR will form resistosomes that go to the plasma membrane, but NRG1 will actually go to different organelles as the cell biology indicates, including chloroplast and mitochondria. So maybe these are organelle busters that are expressing plant immunity through acting at a different site than your typical plasma membrane receptors. Okay, so that was story number one. Story number two is also a kind of unusual application of AlphaFold. And this is something we done in my lab and recently published a paper of using AlphaFold as a way of discovering atypical uh, assembly, atypical protein complexes. And the idea here is that if we know a protein family very well, can we use AlphaFold to identify or predict what are unusual members of that family? What are atypical members of that family? So what we did in this case is we used AlphaFold 3 uh, and develop an index that we call SNI, Structure Novelty Index. This index calculates a lot of parameters about the structure itself. And this was used to identify atypical NLR resistosomes. So I'm gonna skip this, but I'll show you the data in a little more detail here. So basically, what we have is this is the family of proteins that have this hexameric resistosome I just showed you, and this is a big family as you can see. And in this family, there are three structures, NRC2, NRC3, and NRC4, that were experimentally determined as all being hexamers. So six of them come together and form this resistosome. So you can imagine that with this result, it's very difficult to convince someone in the lab to go and try to solve experimentally another one of these structures because three members of this family are hexamers. It's very easy to assume everybody is a hexamer, right? So why should you spend a lot of effort solving the structures, additional structures from this family, right? To rediscover that's a hexamer. So this is where this SNI turned out to be incredibly useful. So what the SNI is, Structure Novelty Index, is basically a bunch of parameters which are listed here on the left. And these are different parameters. For example, the length of the funnel, distance between different motifs, distance between the protomers. So, are they close to each other or not, etc., etc. And these are basically a bunch of parameters that we developed, and then we applied to the whole family. And what you can see here is the ones that have experimentally solved structures are shown in red. So you have an RC3, an RC2, and an RC4. And if we cluster them, they all cluster together. So presumably, all of these are likely to form hexamers because their structures, the AlphaFold models, are similar to the non-hexamers. However, we have these guys here that are not clustering with the non-hexamers. They came out as outliers, right? And so now what we're doing here, we're using AlphaFold to generate hypothesis because AlphaFold is basically telling us these proteins here are deviating from the models we predict for them are deviating from the hexamer. So they're different, whatever they are. It didn't AlphaFold not tell us exactly what the structures were like, but it told us they are different. And indeed, we prioritize these for experimental work. And one of them, so as an RC7, let me go back here. So this is an RC7, which was very different from the known structures. That turned out to be a completely unexpected resistosome with 11 protomers. You can see here this is a big wheel, and there are 11 instead of the six that were for the previously solved structures. So you can see how AlphaFold here was incredibly useful, not in telling us what is the correct structure, but in telling us that this protein is very likely to form a different oligomer than your typical one. That's why we call it structure novelty index in the sense that what is deviating from known structure, right? And we went on to test this for other members of this family, and turns out all three members of this NRC7 family that we tested all have 11 MERS. And here you can compare it to typical hexamers. So this guy here has six. So you can see how much smaller it is and how divergent it is really from the NRC7. But when you try to model NRC7 as a hexamer, AlphaFold say, no, no, I cannot do this guy as a hexamer. It must be different, right? So this is another application of AlphaFold, and frankly, one that really impresses me because I really doubt that we would have spent a lot of effort chasing additional structures here if we didn't have this hint from AlphaFold that we should be prioritizing certain members of this family as being atypical. There's another application of AI in this project that popped up actually kind of unexpectedly as we were doing this project. And basically, the application was that at the moment we were involved in this project, we started collaborating with Google DeepMind, and they developed a tool called Co-Scientist. And Co-Scientist is designed to help scientists design hypotheses, experiments, etc. And so we said, okay, this is a great example to test the system. At that time, of course, we didn't publish the SNI. So we just basically fed the same problem to co-scientists and say, okay, we want to find atypical structures. These are the three known structures. Help us find new structures. And co-scientists basically develop also an SNI similar to the ones we developed. It was not identical. It actually came up with parameters that we identified as human scientists. It proposed similar parameters, but also proposed different ones. So it was not exactly identical to our approach. But what's interesting is both our approach and the co-scientist approach, they both would have identified an RC7 as outlier, as atypical. So if we did this 100% with an AI system, we would have also discovered an RC7 as being atypical. So that's kind of a glimpse into the future where now we have even AI systems that can actually generate hypotheses, generate even strategies, and tell you exactly how to approach a particular problem. And the co-scientist paper is now published. You can actually look at it, and we're quite proud that we were one of the first labs to actually use it for an application and actually even test its performance compared to basically human scientists. And as I said earlier in the introduction, I think before Najib came, but the point here is not whether co-scientist is performing better than human scientists. We both performed similar levels. Each had sort of different parameters, but was not identical. But the point really here is the value of the AI system. It's scalable. We can actually now apply this approach over and over again much faster than if you have a human scientist sitting there looking at the structures and determining what the parameters are. So this can be automated and scaled. And that's really the value of AI systems, is it really accelerates and enables you to automate a lot of these analyses that normally a scientist will have to do themselves. Okay? So that's kind of the conclusion really from this, is that SNI is a scalable framework to enable discovering new assemblies and so on. Okay, so that was my second example. Again, you can read about it. You can also read about co-scientists. And my final example is actually something that is still in progress. So I don't have results for this. But this is a project that we have in the lab that is funded by Google, by google.org. This is the philanthropic branch of Google. And this is a little bit a dream we have in the lab, which is, can we predict disease resistance genes from solely starting only with plant and pathogen genome sequences? If you give the scientist or the co-scientist or the researcher a plant genome and a pathogen genome, can they predict which one, with what is the interaction there between the two, and what is the resistance gene? Or can you even interpret what plants would be resistant based on just the genome sequence? And this is something we're using. We're applying AlphaFold to predict protein-protein interactions here between particular domains in these receptors that are known to bind pathogen effectors and pathogen effectors. And this is, again, this is the project that the lab decided to call it Bifrost. Don't ask me why, okay? So it's, you can ask Amir Ali why he called it Bifrost. But basically what we're doing here is we're predicting protein-protein interactions between pathogen effectors and domains of immune receptors. And at the moment, there's like a large number of these models have been generated, 750,000. And I have to say that the validation is still in progress and is not very straightforward. So it's not, I don't think these tools for protein-protein interactions are yet ready for prime time or for really becoming very routine. I think there's still some improvements that need to be made. But anyways, I want to alert you to this because I think this is the future and this is already happening. And I think we're not very far from the moment where this predicting PPIs, protein-protein interactions, becomes very routine way of exploiting genome sequences, all the way to predicting networks and interactions. And maybe even as people now are dreaming, building virtual cells using this kind of approaches. So that's all I had. So just to kind of recap on the three examples I gave you. So these are problems we were kind of stuck with, maybe still stuck with, and AI had broken some of them, may break another one. So the first one is NRG1. This is this NLR that targets organelle membranes. We AlphaFold gave us a lot more confidence about the mechanism and also helped guide experimental approaches. The 11-mer, so the atypical resistosome that breaks the pattern of hexamers, and this was revealed by this structural novelty index applied to AlphaFold structures. And I think this approach, SNI, is applicable to many protein complexes. If there is prior knowledge on structures, then it can be applied to find atypical protein structures, protein complexes, exploiting again the diversity of genome sequences we have. And finally, my dream is to predict disease resistance genes from genomes only, and that's still ongoing. We haven't broken that one yet. So yeah, so with that, I'll stop. Just want to thank the lab, but you'll be meeting them in the coming weeks, and I'm happy to talk to you. And we post regularly on the blog articles that are maybe more accessible about these different topics, like the mokusatsu I told you about. So I don't know if you have any questions, but I'm happy to chat further with you. So.